The prospect of human cloning has long captivated the human imagination, straddling the line between scientific possibility and fantastical speculation. From the earliest days of science fiction, the idea of creating a genetic duplicate of a human being has been both a source of wonder and a cause for concern. It has been the subject of countless science fiction novels, films, and heated debates, often portrayed as a harbinger of dystopian futures. These stories frequently depict a world where cloned humans are used for nefarious purposes, such as organ harvesting or creating a subservient class of people. But, how much of what we think we know about human cloning is based on scientific fact, and how much is rooted in fictional narratives? The line between reality and fiction is often blurred, leading to misconceptions and fears that may not be grounded in actual science. This essay delves into the intriguing world of human cloning, exploring the science behind it, the ethical dilemmas it presents, and the potential ramifications for the future of our species. We will examine the current state of cloning technology, the potential benefits and risks, and the moral questions that arise when we consider the possibility of cloning humans. The idea of creating an identical copy of a living organism, once confined to the realm of mythology, has inched closer to reality with advancements in biotechnology. Techniques such as somatic cell nuclear transfer have made it possible to clone animals, and researchers are continually pushing the boundaries of what is scientifically feasible. While the successful cloning of animals like Dolly the sheep made headlines, the question of whether humans can or should be cloned continues to spark controversy and ignite passionate discussions about the very essence of human existence. The ethical implications are vast, touching on issues of identity, individuality, and the potential for exploitation. As we stand on the brink of this new frontier, it is crucial to engage in thoughtful and informed dialogue about the future of human cloning. The debate is not just scientific, but deeply philosophical, questioning what it means to be human. Can a clone truly be considered an individual with their own rights and autonomy, or are they merely a copy of someone else? These questions challenge our understanding of identity and personhood. As we explore the potential of human cloning, we must weigh the scientific possibilities against the ethical considerations. The future of cloning holds promise for medical advancements, such as the potential to cure genetic diseases, but it also poses significant moral and societal challenges. The journey into the enigma of human cloning is just beginning, and it is a path that requires careful navigation. At the heart of cloning lies a scientific technique known as somatic cell nuclear transfer, or SCNT. This groundbreaking method has revolutionized our understanding of genetics and cellular biology, offering a glimpse into the potential future of medical science and biotechnology. This complex procedure involves replacing the nucleus of an egg cell with the nucleus of a somatic cell, which contains the complete genetic material of the organism being cloned. The somatic cell, which can be any cell from the body, except sperm or egg cells, carries the full DNA blueprint of the donor organism. By transferring this nucleus into an egg cell that has had its own nucleus removed, scientists create a new cell that has the potential to develop into a clone of the original organism. The reconstructed egg is then stimulated to divide and develop into an embryo, which is implanted into a surrogate mother. This stimulation often involves a combination of chemical and electrical signals that mimic the natural fertilization process. As the embryo grows, it undergoes the same stages of development as a naturally conceived embryo, eventually forming a blastocyst that can be implanted into the uterus of a surrogate mother. If the process is successful, the surrogate gives birth to an offspring that is genetically identical to the donor of the somatic cell. This means that the cloned animal shares the same DNA as the donor, making it a true genetic copy. This technique has been successfully used in various animal species, including sheep, cows, and even endangered species, providing valuable insights into genetics and development. While the science behind SCNT is well established, the successful application of this technique to humans remains elusive. The process is fraught with technical difficulties, including low efficiency rates and high rates of abnormalities in cloned embryos. These challenges have made it difficult to achieve consistent and reliable results in human cloning attempts. Despite claims and counterclaims, there is no credible scientific evidence to suggest that any human clone has ever been brought to term. Many of the reported cases of human cloning have been debunked or lack sufficient scientific validation. The scientific community remains skeptical, and rigorous peer-reviewed research has yet to confirm the feasibility of human cloning. The technical challenges associated with human SCNT are significant, and the ethical implications of such an endeavor are profound, leading to widespread apprehension and regulatory restrictions. Ethical concerns include the potential for exploitation, the welfare of cloned individuals, and the broader societal impacts of human cloning. As a result, many countries have implemented strict regulations or outright bans on human cloning research, reflecting the complex interplay between scientific possibility and ethical responsibility. Over the years, there have been numerous claims of successful human cloning, often capturing global attention and sparking media frenzies. These announcements, whether true or false, have always managed to stir public curiosity and debate. The idea of cloning humans touches on deep ethical, moral, and scientific questions that intrigue and sometimes alarm people worldwide. However, most, if not all, of these claims have been debunked as hoaxes or lacking credible scientific evidence. Scientists and researchers have repeatedly pointed out the lack of verifiable data and reproducible results in these cases. The scientific community remains vigilant, always ready to scrutinize and challenge any new claims that arise. One such example is the case of Eve, a purported human clone announced by the Raelian movement in 2002. The Raelians, a religious group that believes in extraterrestrial origins of life on Earth, claimed that Eve was the first in a series of planned human clones. This announcement generated significant media coverage and public interest at the time. The group claimed that Eve was the first in a series of planned human clones, but they never provided any scientific proof to substantiate their claims. Despite repeated requests from the scientific community and the media for evidence, the Raelians failed to deliver any verifiable data or peer-reviewed research to back up their extraordinary claims. Such sensationalized claims, often driven by a desire for notoriety or financial gain, underscore the importance of critical thinking and relying on verified scientific evidence when evaluating claims about human cloning. The allure of being the first to achieve human cloning can lead individuals and groups to make bold, unsubstantiated statements. This makes it all the more important for the public to remain discerning and for scientists to maintain rigorous standards of proof. It is crucial to approach such pronouncements with a healthy dose of skepticism and demand rigorous scientific scrutiny before accepting them as fact. In a world where information can spread rapidly and misinformation can easily take root, maintaining a critical mindset and demanding solid evidence is more important than ever. Only through careful examination and scientific validation can we separate fact from fiction in the realm of human cloning. The prospect of human cloning raises a Pandora's box of ethical concerns that touch upon our fundamental values and beliefs about human life, identity, and dignity. Opponents of human cloning argue that it is morally wrong to create human beings through artificial means, potentially subjecting them to psychological harm and societal stigma. They fear that cloning could lead to the commodification of human life, reducing individuals to mere copies of their genetic predecessors. The potential for exploitation is another major ethical concern. Critics argue that cloning could be used to create designer babies with specific traits, exacerbating existing social inequalities and undermining the intrinsic value of human diversity. The possibility of using clones for organ harvesting or other exploitative purposes raises further ethical red flags, highlighting the need for robust regulations to prevent such abuses.